guys. So anyway, yeah, thanks for the opportunity to talk about our environmental DNA work. I'm going to present some of the work on the adult or post-settled COTS eDNA that we do. Hang on a minute. There we go. And so, again, just an outline. I'm going to spend just a small amount of time going through our adult eDNA workflow, just to put everyone in that sort of brain space. I'll then talk a little bit about some of our proof of concept work, and then launch into what we've been doing now, and then hopefully just sort of pull it all together with where we want to go in the future and some aspects around making this an operational method. And so, thanks Ben for the previous talk. He gave the definitions of eDNA, so I won't bore you with those. But as far as the sample goes, for the larval work, the eDNA sample is a plankton tail, as Sven said, which concentrates typically several cubic metres of water down to a very small volume. For the post-settled or adult work, we are filtering just a couple of litres of seawater. Now, the reason I'm saying this straight off the bat is that one of the first questions we get is how can we distinguish between the two? And again, as Sven sort of alluded to, the way we distinguish the two samples is by time. So the plankton sampling occurs, or the eDNA work for the plankton sampling occurs primarily in the summertime when we're spawning, where the post-settled or adult work occurs out of spawning, so typically between May and October. So again, just to orientate everyone for our workflow, it begins by collecting a surface water sample through a membrane filter. And this membrane just captures things like cells, mucus, and other sort of biological tissue, like Sven had mentioned, that released into the water. From here, the filter undergoes some DNA extractions, and the resultant DNA extract contains a soup of DNA, from all of the organisms that are in that water body. And for our COTS work, this DNA is then analysed using our COTS-specific genetic markers. And so this generalised eDNA workflow is actually quite typical around the world and embedded into a lot of regulatory frameworks already. So I'll step back to some of our previous work just as for our proof of concept. So we visited about 11 reefs during 2016 and 17, some that were undergoing outbreaks, such as Perth and Rib Reef off of Innisfail, and then others that were not around Elizabeth Island up in the north, and at the time Davies Reef near Townsville. And so during these trips, we did some visual transect surveys and then collected underwater samples, as well as surface water samples, because we wanted to see if there were any differences there. All of those eDNA samples were then extracted, and that DNA soup that we extracted was then analysed for COTS eDNA specifically. So this plot on the left here is the COTS density from all of those sites. Pretty straightforward. The larger the bubble, the higher the number of COTS that we saw. We're happy to see when we overlay some of our eDNA results, we got some fairly comparable data there. And so this plot on the right here is actual eDNA concentration from the underwater samples. So we also looked at the percentage of positive samples, so as another metric, because a presence-absence test, as Sven had mentioned, is something that we've worked on in the past, and for the larval work, concentration is important because we can then work back to actual larval densities. For the adult work, we're not finding concentration is as important, and presence-absence assays are actually giving us just as good as information. So putting this data together, and then given that sampling was not controlled for environmental variation at the time, so these linear models, in these linear models, COTS density actually explained a high amount of variance in the COTS eDNA for the samples taken both underwater and on the surface. So these particular linear models are from the ones where we looked at the percent of positive samples, but they're actually quite similar for the ones where we looked at the concentration as well. And so again, just to highlight the fact that it's in a workflow situation, it's really important to make that distinction between whether you're measuring sort of a how much or whether you're measuring presence or absence. Obviously, a presence or absence assay is a much more simpler workflow to integrate. Also, the surface water sampling was something we were pretty keen to investigate, as obviously that's going to be a preferred collection method going forward. Just to sum those two up, from concept work, we're very happy to move in a direction that involved surface water sampling and presence-absence assays. So now we'll fast forward a little bit. We've got several years of post-settled adult eDNA data and have done a lot of refinement for sampling and measurements and how we do things. What we know from this previous work is that at reefs undergoing outbreaks, it's actually quite easy to detect COTS eDNA in just a couple of litres of seawater. We are interested in developing this as an early warning tool such that we can identify reefs that are below outbreak levels and undergoing shifts in population densities. And so we've heard that from others already, that the need to act as soon as we identify these population shifts is quite paramount. What I'll show you now is some 2020 data. And these are just some 4 locations that we've been to quite regularly, but this is just the 2020 data. We've got Lizard Island, Davies and Bowden Reef off of Townsville, and Elizabeth Reef off the Whitsundays there, all of which were well below the Manto threshold, no outbreak, definition of 0.1 COTS per toe. So we wanted to use Elizabeth Reef, which I think Mike had presented in his previous slide, presentation as well. Thanks for doing that survey, Mike, as well. It was very handy. We did some surveys in 2019 as well and didn't see any COTS at Elizabeth Reef. So we actually were quite happy to use Elizabeth Reef as a background reference, you know, to work towards this baseline for our eDNA signals. And so we collected a large number of samples from each of these locations. I've just brought forward that Manto data from the previous slide as well, just to, and again, just highlight that they're all well below that 0.1 COTS per toe level there. And so in its simplest form, these pie charts pretty much represent the number of positive and negative samples in each of these locations. And you can see that the proportion of positive samples actually aligns quite well with the Manto data there. And so from this information, we can use some statistical modelling to determine the probability of a sample being positive, and then determine the number of samples that we need to take at these kinds of locations to ensure we're operating at a high level of confidence for detection of positive samples. So for places like Davies and Bowden Reef, it turns out that you know, barely a few samples is all that's required to ensure you've got that level of confidence that you're seeing a positive sample. Lizard Island, it's more like 10 to 15 samples, and then at a place like Elizabeth Reef, again, looking at that as a potential baseline signal there, we're sort of around the 40 sample mark. But what this information gives us is, it enables us, is to identify maybe a fixed number of samples that can span all the possibilities of these no outbreak reefs. Now, one of the questions we often get asked is, how do we know that the COTS eDNA we detect actually comes from that reef? So we've gone about it trying to answer this question. In doing so, we need to consider how the COTS eDNA gets into the water and what happens to it. So in the eDNA field, the term shedding is often used to describe this release of cells and mucus and other biological tissue from the organism of interest. And so that's much like when we wander around and we lose hair and skin and all sorts of things around us. So once this biological material is in the water, they undergo a variety of mechanisms to reduce their concentrations. The primary mechanism are either a decay of some sort or via transport, so relocation of that material to some other place. So firstly, if we start looking at the decay side of this story, what we know is that COTS actually shed eDNA at a very large rate. So in fact, many millions of DNA copies are released every hour by every single animal. And recent studies by a student of ours and Morgan's, Sarah Kwong, has revealed a rapid decay rate of the COTS eDNA, such that in the absence of continued shedding, any COTS eDNA produced is actually completely gone within 4 days. Now, the other aspect, as I was saying, is transport. And then what we wanted to do to investigate this aspect was do a transect across a reef with known COTS populations. So to do this, we went to Keeper Reef in 2020, remembering that Keeper Reef is one of those that is under control by USO and his team at PMG. So in front of Keeper Reef, all of the samples we collected there were negative for COTS eDNA. The samples that were collected right on the reef, 80% of those were positive. At our anchorage, just a couple of 100 metres behind the reef, only 30% of those samples were positive. And then if we go sort of a conglomerate of half to 2 nautical miles behind the reef, all those samples together, only 2% of them were positive. So bringing these two pieces of information together, that is the rapid decay of COTS eDNA and the dilution to transport of eDNA off a reef, we're pretty confident that any COTS eDNA we detect originates from the reef from which it was sampled. So in other words, COTS eDNA detected at a reef is not likely to have originated from an adjacent reef. So with all of this, we feel like we can start focusing our efforts on operationalising this eDNA method. And what this means is pulling apart the typical eDNA workflow that I showed at the beginning and putting it back together in a way that can leave us with multiple options for simplified workflows. So, for example, we've been testing this recent product called a self-preserving filter device. And what this does is once you filter the water, you can simply put this back in the bag it came from and the inbuilt desiccant just dries out the filter and preserves the DNA. The testing we've done so far is that the DNA is just as good as it was from the day it was sampled. At least 3.5 months later, we're actually taking that testing out to 6 months. The good thing about this is that disseminating an eDNA sampling program to external collaborators using this kind of approach actually becomes a very, very achievable option. As another example, we've recently developed a COTS dipstick which sits at the detection end of the workflow. And so this form of detection requires no technical expertise or laboratory equipment. It's just a visual interpretation and provides very sensitive test to detect very, very small amounts of COTS eDNA. Now, both of these were going to be demonstrated tomorrow. I guess that's not going to happen for all sorts of reasons. So if anyone wants to talk about these at another time, feel free to drop me a line. So going forward, again, we anticipate we're going to have a toolbox of options rather than just a single method. So things like this new filtration apparatus are probably ready to be rolled out almost immediately. We've got a number of other options for on-site analysis coming online, should that be an approach that is needed. And also, we're going to try and expand the number of non-outbreak reefs under survey just to increase our confidence in these baseline eDNA signals and certainly shifts from this baseline. Last but certainly not least, we're looking at these e-reefs models to see if it's possible to identify site-specific sampling locations. So, for example, water residence times might indicate locations where we could expect concentrations of eDNA, and then they might become preferred sampling sites. Excuse me. So just in summary, we present an eDNA as a reef scale early warning method. Just to be clear, we're not advocating eDNA replaces current monitoring methods, rather it might be a fit-for-purpose application. We've already, oh, excuse me, bit of a cold still. We've already heard about some of the workplace health and safety issues around mental towing and the importance of identifying these population shifts in non-outbreak times. So I'll definitely fly the flag for eDNA monitoring as a sensitive tool. And in fact, I might, if it's okay, stop there because I'm just about to have a large coughing fit. So I'll put myself on mute and stop right there.